good morning, folks. It's 3301 once again. Here we are. We're enjoying the time change. We're enjoying our fall semester. We love it, don't we? The topic for today is Fragile X syndrome. Let's jump right in. So here's the title slide. Fragile X syndrome. This is the second of our two-part series on trinucleotide repeat disorders, the first being Huntington's and the second being Fragile X. And then later this week, we'll talk about autism spectrum disorders, which, though not classically seen as a trinucleotide repeat expansion disorder, do have an element of repeat expansion in the kind of the latest and most modern understandings of their genetic etiology. So 3 lectures kind of put together in a fun and informative way. Now, it brings me no pleasure to announce this, but we do in fact have another midterm coming up. It's November 16th, so that's 1 week from today. The format is going to be the same as the first midterm. So the first midterm had 20 questions. They were set up to be about 1/2 written and 1/2 multiple choice. Multiple choice in this case, not always literally multiple choice, but it could be fill in the blanks or matching or something along those lines. Anything that can be machine graded essentially would fit into the multiple choice category. And so yeah, I mean, if you felt that you learned something about how things worked from the last quiz, you can bring that to the current one and hopefully do reasonably well. I wanted to just give you some starting points on what to focus on for your studies. This is not to say that any or all of these topics will be on the exam. Of course, that would be foolish for me to give away the exact content. But these are the kind of things, just going over my slides, that I think I would be tempted to include in some capacity. Because, for the written questions especially, I like to ask sort of bigger open-ended questions, sort of, explain, for example, why cellular heterogeneity is a confound, why it is that you can sometimes run into problems when you're studying gene expression or epigenetics in bulk dissections of brain tissue. And you can go into the whole part of the neuroanatomy lecture that we talked about that. There might be some other parts you could bring in from other lectures that we'll get to, where we also show how researchers use cell sorting to cope with the problem of heterogeneity. But there's a lot of things you could kind of ask about that. Another broad one would be how are SNPs and genomic regions linked to functional genomics. So this is a topic that comes up fairly often whenever researchers do a GWAS study or generate a DNA methylation data set or a ChIP-seq data set or something like that. They always want to map those features onto known functional annotations. So for example, they might ask, what do our SNPs that we found map to any enhancers? Do they map to promoter regions, or what kind of context do they map to genes that are expressed in the brain? Just the general concept of taking the GWAS and genetic results you get and trying to contextualize them with as much other information as you can think of. So that's something that we talked about in a few separate lectures, and so there's a way I could perhaps think of a question that tries to bring those together and asks you to pull from multiple lectures examples or a description of what researchers are doing. So something to think about there. Simple one also would be a question about heritability. A lot of things that could be asked about that because we took a few slides to talk about that topic. Linkage has been a common theme in many lectures. What is it? How is it? What causes certain regions of the genome to be linked? How is it measured? How is it used in things like, how is it used in the precursors to GWAS studies, the linkage studies? A lot that could be asked there. There's some nice juicy material talking about DISC1 and the major histocompatibility complex. There, in the schizophrenia lecture, I really tried to kind of stretch out and just give you a longer, kind of more historical viewpoint on how these things were discovered and how they've been linked to actual morphological or histological differences in the brains of individuals with schizophrenia. So there's a lot that we could kind of work with there. MQTLs and EQTLs, what are they? How do they impact our understanding of epigenetics in the brain and disease? Repeat length expansion, that of course is a theme that is covered by this lecture as well as the previous one, as well as to a certain extent the ASD lecture. So you could imagine a question that tries to unite all 3 lectures and say, I don't know, it's hard to say, but like, how is it detected? Why is it challenging to detect? Why is it that repeat lengths are able to expand so easily, et cetera, et cetera, et cetera. Lot to be asked there. Somatic mosaicism is another topic. We may, that might be something that might be a bit better for the final, because I think there's a little bit more I'd like to tell you about that in the last sort of set of lectures. But there's something to be said for somatic mosaicism, the tendency for the individual cells in our body to have somewhat different genomes as a result of mutations that pile up throughout our lives. And then questions about genetic correlation and overlap, that will be covered tomorrow. And I think there's but we also talked about that in the schizophrenia lectures. So there's one of those examples of a concept that comes up in multiple separate lectures that I'd like to ask you a question about that sort of unites them. So these are just starting points. I might also ask you to interpret figures. I haven't made the exam yet, so I can't be super specific about this, but these are just some of the ideas that I thought of as I was going through the material this morning. But yeah, I could potentially ask you to interpret a figure. I might pull one up from something that is visually or thematically similar to something that I've told you about in class and ask you to interpret it. That's possible. I don't know yet, but it's one of the things that's on the table. Yeah, I think that's about it. And as I've said a number of times, I think part of what helps you study is trying to get into the mindset of your instructor or your professor and think about the way they would approach writing the questions. Because ultimately, that is where the questions come from. I have to look at the material and think of them. So you kind of have to think, okay, well, if I was Harry, what would I want to ask about that gives enough of a challenge and covers enough of the material to make it seem like the class is kind of hanging together and has a consistent narrative. So you can kind of think about that. Another thing you can do is give yourself little starting phrases that kind of lead you off. So you might look at for example, the 2 schizophrenia lectures, and you might ask, okay, well, if you were to start a question with compare and contrast, what would I be asked to compare and contrast? Or if he were saying like, what are the different types of blank? Or, discuss the implications of blank. You can kind of give yourself little verbal starting points that are typical or kind of common motifs in a lot of exam questions. And then once you have those, you can think about, okay, well, realistically, what could I be asked to compare and contrast in this context? And so you can sort of think of that type of thing. So so that'll be, so I'll give you that as a starting point. Hopefully on the next class, I'll have actually come up with some of the questions that are going to be on the exam. And I guess I can, I won't tell you what they're going to be, of course, but maybe I'll have more specific insights. I'll also have to go through and look at the YouTube analytics for the lectures that we have done, because those analytics do, if the videos have enough views, I'm able to see where it is that students keep coming back to. So I can kind of zoom in on parts of the lectures that people seem to watch repeatedly, presumably because it's something that they think is hard to understand or important and worth concentrating on. So I can kind of like use the power of YouTube analytics to figure out the parts of the lectures that need that need more focus. So that's kind of a fun thing to do. It only works if enough people watch the videos, though. So for the newer ones that haven't gotten a full number of views yet, the possibility may not be there. Speaking of that, I think I guess I would urge the people in this class to try to stay on top of the videos, the video lectures. I know it's not, perhaps not realistic to expect everyone to want to wake up at 8:30 in the morning and come to the class live, but I think that the number of views that we get on the videos that have been posted still aren't really consistent with the number of people in this class. So it seems to me that many people in this class are not watching the lectures, at least not watching them on time. So I think that's going to be a big problem for you if you plan on not failing the exam. I don't know exactly what your sort of workflow is or how you like to get topics done, but if you're not on top of things and not watching lectures actively week by week, roughly around the time they come out, you are really not going to be performing well in the class, to say the least. So if you're having trouble following the material, or if you're having trouble staying motivated, that is certainly a cause for concern, something that would be worth contacting me or contacting your TAs if there's any way we can help you on that front. But if it's just a matter of putting the videos off and putting off the lectures until the last minute, well, I don't need to tell you this, but that's really not a great way to do it, folks. It's not going to get you you're not going to get the results if you do things that way. So my little warning to you, but hey, it's your life, it's your choice. So all I can do is give you the grades. So let's get into Fragile X syndrome. This is a leading inherited form of intellectual disability and autism spectrum disorder. It's characterized by a range of phenotypes, including primarily poor language development, seizures, and behavioral alterations like hyperactivity, impulsivity, and anxiety. There's also a fairly prominent physical anthropometric phenotype. Broad forehead, elongated face, large prominent ears, occasionally crossed eyes, or strabismus as it's called in the literature, and various other kinds of things. So it's a condition that has both cognitive and physical attributes. And it affects about 1 in 5,000 males and between 1 in 4,000 and 1 in 8,000 females, although the exact numbers differ depending on who you ask, but it's somewhere in that neighborhood. Both males and females can have it, although females typically have milder cases. The reason for this is that females, of course, have 2 sets of X chromosomes, whereas the males only have 1. So if you have this Fragile X mutation, if you're a male, you have no spare chromosome to kind of smooth out the rough spots. But if you're a female, you do have a spare X chromosome, and so typically the phenotype is not quite as severe. Sometimes females can be carriers of Fragile X and not even really know it. You might be able to measure a difference in intelligence at in a very precise way, but it may not be enough to notice in the day-to-day life. Oh, so we have a question in the chat. So Natalie asks, quick question about the midterm. The quiz will be on November 16th at 8:35 a.m. Yes, I think that's the plan. It's whatever it was last time. So I think I had it start at 8:35 in the morning. And then it's open for 24 hours as they all are. So let's see here. I'll just look it up because I don't want to go on record saying something that's not consistent with the syllabus. Yes, so the quiz will open at 8 a.m. on November 16th, and then it will, November 15th. What does that mean? Is that right? Oh yeah, it's November 15th. Sorry, everyone, it's due November 16th. The quiz is on November 15th. So it opens November 15th at 8 a.m. and it closes at November 16th at 8 a.m. So that's on the syllabus. The syllabus is king. Don't forget that. I just made a mistake on the slide. So yeah, and it's worth 20% as always. Sorry for the mistake. I don't know why I wrote 16th. I was kind of looking at the time or the date today. It's like the 8th. How could next week be the 16th? And then I thought, well, maybe the weeks have turned to 8 now. That'd be nice if we could get an extra day in the week. So anyway, so this is Fragile X, and there are, like I said, a number of physical features. And the most common cognitive symptoms of Fragile X include developmental delays. People would miss their normal motor or language milestones. You get intellectual disability, typically manifesting as lower intelligence, of course, and learning disabilities on top of that. So sort of a whole range of impairments. Now, in the brain, you'll see one of the aspects of the physical phenotype is macrocephaly, which means like large head or large brain. This is not because the brain is like physically filled with more neurons, but in part it is. Some parts of the brain are enlarged. The caudate nucleus, for example, part of the striatum is enlarged. The fourth ventricle is enlarged. That's the ventricle that's located between the cerebellum and the pons. And the hippocampus is also larger. So certain parts of the brain actually are in fact bigger, although bigger is not always better. I think efficiency is an operative word here. If you have extra neurons that aren't contributing meaningfully to the ongoing information processing in the brain, that's not actually going to be good for you, even though they're extra. And decreased lateral ventricle volumes. So the gray matter in the brain in some subcortical regions has actually expanded and actually pushed the lateral ventricles to be smaller. And the extent to which this happens is correlated with IQ, so that the more out of the normal range these volumes are, the lower your intelligence quotient tends to be. And the range goes from mild to severe, but about half of the females with the full mutation Fragile X syndrome gene do have about a normal intellect. So like I said, the presentation and the phenotype is a good deal less severe in females. So here's the clinical trajectory of Fragile X. In infancy, you see sort of, I guess, what you might broadly describe as failure to thrive, problems with digestion and suckling, poor muscle tone, that's hypotonia. Motor delays begin to appear around about the time you start to see motor development. I mean, when babies are first born, they're kind of like they're like Furbies. They just kind of move all of their limbs randomly, and there's not any sort of goal directedness to it. But as they start, as their motor system is called on to do more and more, that's when you really start to notice delays. By the time they start walking, you'll notice seizures and hyperactivity. Around about this time is when language should start to be developing as well, and so you start to notice language delays, anxiety, hyperarousal, otitis media, that's this ear infection. The early childhood is characterized by impulsivity, symptoms of autism, poor eye contact, tantrums and anxiety, and just all around behavioral challenges, and so on all the way into adulthood. And by the time the individual is aging past adulthood and entering kind of the senior, middle age and older, you start to see, in some cases, even further cognitive decline and Parkinsonism. So this also has a neurodegenerative component as it evolves. Now, you might think, or I suppose you might, based on, if you were a doctor, say, in the 19th century or something like that, you would probably see a lot of people coming through with different kinds of cognitive impairment and different kinds of motor problems and different kinds of disturbances all around. And it wasn't really until people started looking at pedigrees and trying to understand things in the context of inheritance that we could really start to categorize the different kinds of cognitive, the different forms of cognitive impairment. Historically, it was not terribly well categorized, and it was in the 19th century that clinicians started to really define symptoms into syndrome was based on a cluster of symptoms. Syndrome just means when symptoms occur together. So here is a pedigree chart by Martin and Bell. They were English physicians in 1943, and they were looking at the pattern of inheritance of this pattern of cognitive impairment seen in a English family. This family had an unusually high number of what they called mentally deficient sons who were born to what appeared to be otherwise normal mothers. And so they drew out this entire pedigree going back many, many generations. And so here, upward arrows in the chart are males and downward arrows are females. Filled circles are cases of full-blown cognitive impairment, typically in males. So what they would have called, they called that imbecility. This was like the official medical term for cognitive impairment in those days was imbecility. So somebody who had this would be an imbecile. Imbecile actually just means like without a stick or like without a supporting staff. Baculum is Latin for stick, and im would mean like the negation of that. So it's somebody who's without a supporting staff, and it kind of is closer to meaning physically weak so much, but typically, or it's not unusual, I guess, for symptoms that cause for syndromes that cause cognitive impairment to also cause some level of physical weakness or physical impairment as well. So that's what the word imbecile means. Now, we use it as kind of an insult these days, but it was at one point a clinical word. Now, yeah, I mean, I think this is kind of the inevitable fate of all of the scientific terms used for cognitive impairment, is that they inevitably just get transformed into insults at one stage or the other. So I think, historically they've cycled through a lot of them as they've tried to refine their terminology and move away from things that have taken on a very offensive connotation. This will probably continue forever. I think I don't know exactly when this could reasonably stop, but of course we know in our own lifespan, certain words have become strongly disfavored because of the insulting connotations they carry. But this is, ongoing throughout history. But anyway, so you can kind of track this family and see that, so like quite often you'll see these instances where a quote unquote imbecile, a cognitively impaired male is born from a normal or apparently normal mother. See the mom here, the mom here, mom here, cognitive impairment here, here, and here, and so on and so forth down the line. So you can see, like, if you lay this all out and you're curious about it, you would start to think, okay, this must be sex-linked or linked to the X chromosome or whatever the case may be. It's clearly coming from moms, because in every case, mom is the person, if the mom is in the family, the odds are that she'll give birth to a cognitively impaired son. Another thing I should point out, I guess, is that it's kind of neat to look at the other features on this plot. Like, count up how many people died in infancy or died of tuberculosis. The numbers are quite staggering, which I think gives you a sense of the rather brutal nature of even mid-century English life, where dying in infancy or dying of tuberculosis was a fairly common occurrence, common enough that it had to be noted on the pedigree charts. Imagine a pedigree chart from today having anywhere near this level of fatality. People could put up with a great deal of death, and it just kind of, it just kind of washed over them. They didn't really think about it too carefully. It's a little bit like the way things are now with the coronavirus, is that it certainly made the world more deadly, but people have seemingly adapted to it and just can put up with a higher level of death in their lives than they used to have. And people are very good at not thinking about that sort of thing. Life has been the deadliness of life has sort of gone up and down across history. Anyway, that's the observation. It's a mental deficiency linked to sex. So this is a curious thing, but it wasn't called Fragile X syndrome back in those days. It was just a sort of an observation of a sex-linked mental deficiency. But once this became noticed, people would pick out more and more families like this, and it became clear that somebody had to investigate the cytogenetics of it. So in 1969, Hubbs identified what he called a marker X chromosome, examining karyotypes from a pedigree with sex-linked cognitive impairment. So this is a different pedigree from the one I just showed you, but it's the same kind of thing. You've got these individuals with, as they call it, mental retardation. Again, old-fashioned term, but this is 1969, not the 1940s. So they've changed to a new term, and again, we're using a different term now. So the syndrome would be passed along from moms down and appear mostly in sons. So anyway, so they made some observations of people of these individuals, and they noticed that the X chromosome had a very strange appearance. This is a karyotype. This is when you capture all the chromosomes in metaphase and fix them in place and then arrange them by size, and that's how you figure out, okay, here's chromosome 1, 2, 3, and so forth down the line. Chromosomes go in order of their size, so the first chromosome is the biggest, and the last chromosomes are the smallest, and the sex chromosomes are kind of in between. Well, the Y chromosome is very small, but the X chromosome is a pretty big chromosome. But anyway, the X chromosome is here. In male, of course, he only has one of them, and you can see there's this kind of like weird marker, as they called it, these little tiny satellites that are kind of sticking off the tail of off the long arm of the X chromosome. It's a secondary constriction. Chromosomes do have constrictions around the centromeres. That's a normal part of the way a chromosome is structured. But the remainder of it is not supposed to be very lumpy at all. Here is a constriction that is not visible in control or healthy patients, but is visible in these individuals with the cognitive impairment. So they didn't know, of course, if this was the same syndrome as described by Martin and Bell in the last slide, but it clearly was some type of X-linked cognitive impairment. So what you can call this is a fragile site. It's the name of the condition, of course, is Fragile X syndrome. The reason is that these fragile sites are constrictions in the structure of the chromosome. So the DNA is actually kind of taken on a different three-dimensional shape in and around this fragile site. It's gotten all squeezed in somehow. And under a microscope, this gives it the appearance of being fragile. It's like those little pieces could almost break off and fall away, and then you would lose a big chunk of your X chromosome. So that's what that means by fragile sites, and that's where the name Fragile X comes from. It just has to do with the physical appearance of the chromosome. But after it became known, after this fragile site was known, it became possible in the late 60s to actually study Fragile X syndrome formally, because you could take cases of cognitive impairment and you could look at the family history, and if justified, you could do a karyotype and actually look to see if the proband or the individual that you were looking at actually had this fragile site, and if they did, they were a diagnosis of Fragile X syndrome. You could also follow that back to through their moms and see that, you know, the moms would often have these Fragile X chromosomes too, but of course because they had a spare extra regular ordinary chromosome or a normal X chromosome, the effect of the fragile site was, you know, oftentimes quite minimal. But so there you go. That's it. And once you have an aspect of cytogenetics that's so thoroughly disturbed, you can kind of assume that would cause a lot of serious problems for the individual. But researchers didn't know what caused this fragile site in the first place. And if you're thinking about it from the point of view of the 1960s when it was discovered, a couple of ideas would come to mind. One would be that there is a sequence variation. They have a mutation there that just causes it to be constricted in that sort of fragile looking way. That is one possibility. Another possibility is that it is some kind of inborn error in DNA synthesis or metabolism. Maybe some aspect of how DNA is synthesized is thrown off. People in those days were very into biochemistry. I think I don't know what the impression you get from the courses you take, but if you're taking courses in the neuroscience department mostly, you probably get the impression that biochemistry is not a terribly important thing to understand the brain. And in the modern day, this is actually kind of true, because a lot of the work we do doesn't really get into biochemistry a great deal. The biochemistry has kind of been worked out, and now that we know it, we can go and look at molecular biology and kind of more sophisticated topics. And that's not to say that biochemistry isn't important. It's just that, you know, a lot of what goes on in neuroscience doesn't directly interface with it. It's going on in a different building in a different department. And quite honestly, most neuroscientists aren't very good at chemistry. That's why they became neuroscientists, not chemists, after all. Anyway, in those days, biochemistry was big, and people thought that there was some way that it could solve everything. So there could be some kind of problem in the synthesis of DNA or the way nucleic acids were metabolized or something along those lines. So a great deal of work was put into trying to figure that out. So I kind of maybe go around and just talk about different attempts that were made to kind of figure this out. And one thing that was discovered reasonably early on had to do with the fact, or came from the fact that the fragile site on the X chromosome had a variable expression in cultured lymphocytes. So you can capture, or you can gather lymphocytes from people who are carriers of the Fragile X mutation and culture them, and they'll happily divide endlessly if under the right conditions. And you can look at the lymphocytes and see, does this Fragile X, this Fragile X chromosome, is it fragile in all of the subsequent generations of cultured lymphocytes? If it's preserved across many cell divisions, that tells you one thing. On the other hand, if the fragile site goes away, or if something you can do to the medium that the cells are cultured in changes whether or not the site appears fragile or not, that tells you a great deal about the biochemistry of the disorder. And so back in the 70s, it was discovered that the culture medium that you cultured these lymphocytes in affected the expression of the Fragile X marker, so this little constricted site. What I mean here by expression is not like gene expression, but just whether that trait is visible or not. So if it's expressed, that means that you see this constriction and this fragile looking thing on the X chromosome. And if it's not expressed, the X chromosome looks sort of normal. And that was kind of interesting. So you could try out a bunch of different culture mediums, and they would have different kinds of ingredients in them, and you'd see different results on this fragile site. And then in 81, Patricia Howard Peebles found that the real secret ingredient here was methionine. So culture media with methionine, you would see the fragile site. But if you made a culture medium that was depleted in methionine or other kinds of methyl donors, you would see that the fragile site would not be expressed as the generations wore on. So this is kind of interesting. So this is I think this is quite an interesting thing, because what it suggests is that Fragile X syndrome might involve one-carbon metabolism. Now, we're not going to get into biochemistry, as I've been saying. Most neuroscientists, myself being number one, are not biochemistry experts. That's not, I mean, if I was a biochemistry expert, maybe I would be making millions of dollars in a drug company or something. But I just order kits off the internet and do the reactions that the kit tells me to do. The one-carbon metabolism pathway is, however, fairly important because it is where methyl donors or methyl groups come from, the one-carbon units, where methyl groups come from for things that have to do with methylation, right? So DNA methylation obviously is going to be at the top of the list for us here in this class. That's the thing we think about when we say methylation, although histones can be methylated too. But you also have things like neurotransmitter inactivation. For example, the enzyme catechol O-methyltransferase, COMT, deactivates or begins the process of dopamine breakdown by methylating dopamine. And I think it methylates it to homovinylic acid, but don't quote me on that. But anyway, it adds a methyl group to dopamine and begins the process of metabolizing it and eliminating it from the brain. Methionine is a dietary methyl donor. So the methyl groups that go into one-carbon metabolism that ultimately contribute to DNA methylation and other kinds of things come from the diet in the form of methyl donors like methionine and folic acid and vitamin B12 and these kinds of things. So here's a little diagram of the one-carbon metabolism pathway. You can see how methionine is an ingredient that feeds in, and there's a number of steps in this sort of cycle that we don't need to get into. It interacts also with the folate cycle. But the important fact of the matter is that this produces a lot of important molecules. This one-carbon metabolism pathway produces a lot of important molecules, many of which contribute to epigenetics and DNA methylation in particular. So from our vantage point here in 2021, this hint that the expression of the Fragile X marker had to do with the availability of methyl donors really makes us think that DNA methylation might be involved, because we're looking at a problem of DNA. We know that methylation can change the three-dimensional structure of DNA by influencing chromatin conformation. And we know that the amount of methylation you have depends on the amount of methyl donors available in your diet. So that really gives us a strong hint. But this is the 1970s, and they didn't have, or I guess the 80s now, they didn't really have quite the same level of insight. This was, many, many years ago. 40 years ago to be precise. So I wanted to just give you an excerpt from this paper and read it. She writes, "The mechanism of biochemical control of fragile site expression is not obvious at this time. Any attempt to explain how the methionine effect works would be pure speculation at this point." This is the discussion section of the paper. She's admitting that she's found this but has no clue why it's happening. Methionine is involved in numerous metabolic pathways. Its three major roles are utilization for protein synthesis, conversion to S-adenosyl methionine to serve as the prime methyl group donor, and conversion to cystathionine, cysteine, and other derivatives. Could methionine have a more significant role as part of histones, non-histones, polyamines? So she's kind of speculating. She says, methionine does all these things. Maybe it's got something to do with cystathionine. Maybe it's got something to do with the histones. I think they probably would have known at that point that histones could be methylated, so maybe she thought something like that was involved. Non-histones, non-histones would be everything else. The only two things in your body are either histones or non-histones, kind of a classic kind of philosophical tautology. But anyway, I think it's fun to just point this out because we're here in 2021. We get to look at her great work, but we also get to know the answer. What a time to be alive. These questions that people devoted their careers to, now we can easily look up and we know the answer to this riddle that puzzled these brilliant thinkers from the past. So I think it's nice to think that we're around long enough we're at a time in the world where we can still make meaningful discoveries. We haven't come along too early to ever even know about them, and we haven't come along too late for them to have already been discovered and to be old news. We're at a time when the discoveries are actively being made, and we get to see all of these questions being answered in real time. So I think it's quite nice to consider that and what a time it is to be alive. It's nice to think also that we'll be around long enough that many of the present day questions we have will be answered. This course, I hope, raises many questions in your mind, and the other courses you take would raise many others. And if you stick around long enough, you'll probably hear the answers to many of them before you're gone. So just something to appreciate. Anyway, let's move on. So this is the... So that gives us a hint, I guess, that DNA methylation is involved in this. We'll put that to the side for now and look at the fragile site itself a little bit more. Because methylation can't be the only thing, because you could, if you have a normal X chromosome and you're from a healthy family, the amount of methylation you have doesn't cause Fragile X syndrome. So it's got to be something that's involving a genetic mutation at the same time as something epigenetic. The two kind of have to work together. So back in 91, you et al. Were able to actually locate where the fragile site was on the X chromosome. You could locate it cytogenetically, and you can kind of look at its physical position on the X chromosome and figure out what band it corresponds to and that kind of thing. But that's a little bit short of the molecular precision that you would get from modern day 1991 level recombinant DNA techniques. So here is the pedigree of a family that carries the Fragile X mutation, and anyone who's black is a proband. There's somebody who is a carrier and has the syndrome, and if you're a circle, you would be a female who has one fragile chromosome and one regular one, and has maybe a milder phenotype. But anyway, we're looking at the black people, or the black colored boxes, I should say. The study is... Anyway, the authors of the study were able to, through fairly complicated cloning techniques, develop a DNA probe that could bind to the region of the genome that's actually mutated in Fragile X. Then, using this, they were able to use a technique called Southern blot analysis, which is an old-fashioned way of measuring DNA, basically quantifying the length and sequence position of DNA, and show that the status of whether you were a disease, if you had the disease, or whether you carry the disease, was linked to the length of this particular region. So here it is. Here is an electrophoresis gel, and as with all electrophoresis gel, they're taking a piece of DNA that they've a fragment of DNA, and they're running it along the gel, and the smaller pieces of DNA migrate more quickly and end up toward the bottom of the gel. The higher molecular weight pieces of DNA stay toward the top of the gel under the influence of this electric current. So where you see small pieces, that indicates the length of this X chromosome marker that is normal. And where you see fragments that are up here, it means that there is a longer fragment. The length itself can vary, but they have longer than normal. So everyone who has a fragment down here would be approximately normal. Anytime you see a fragment above that, they would be longer than the average. This fragment would be longer. And so what you'll see is that in, for example, this proband here, this carrier, or this patient with Fragile X, has no band down here in the normal range. His band is all the way up here. It's very faint and tough to see, but his band is considerably longer than what you see in the regular healthy person. Here, this person is a carrier, so they have a band that's up here, but also a band in the regular position. Same for this person. She has a band in the regular position as well as a fragment or band in the in a longer position. And here is another proband. Here's another person with the syndrome. No band in the healthy position and one in the longer position. So what you'll see then is that anybody who happens to have this Fragile X syndrome or carry it on their X chromosome, this particular part of the X chromosome is longer. That's what the gel is telling us, is that when you run it out, it's this section that they've captured with this probe is higher molecular weight and thus a longer stretch of DNA. So we have the idea that there is this expansion of something. And I just wanted to point out that the authors of this paper, Yu et al., back in 91, were talking about this fragment that they identified and said that it's GC rich, so rich in guanine and cytosine base pairs. And it's refractory to PCR analysis for that reason. That just has to do with melting temperature and the nature of GC content being hard to anneal and hard to melt. That's an aspect of PCR you don't need to worry about. But anyway, they know that the fragment has recognition sites for several CpG-dependent restriction enzymes. And what happens at CpGs, folks? Methylation. So they're saying, essentially, that this area that has repeats is filled with cytosines that apparently are able to be identified by CpG-dependent restriction enzymes. So there's a lot of CpGs in this repeat is what she's saying. And of course, this is back in the day when sequencing things was quite challenging. So they use restriction enzymes to kind of identify what the sequence might be. And they imply, or they determine that there's a lot of CpGs there. Sure enough, in that very same year, 1 month after Yu et al. Published their paper, Kremer and company showed, by actually sequencing this part of the genome, that the repeat sequence in the Fragile X restriction fragment contained a CGG repeat. So this is an area of DNA where the letters C, G, and G repeat endlessly. So here is an old-fashioned sequence that they've printed out in their paper. The repeat is underscored here, and you can see it's just CGG, 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 endlessly repeated over and over and over again. And the length, of course, could depend. It could be this long, or it could be, it could go all the way down to here and push everything else out of the way. But it's variable. But it's a trinucleotide repeat expansion with the letters CGG. And of course, CGG, right there at the C and the G, that's a CpG site. So that cytosine there can be methylated. That is a cytosine that's open to methylation. Remember that methylation doesn't typically happen at most cytosines. Most cytosines are unmethylated, except in the CpG context. So you have here a very CpG-dense area that can be expanded by this Fragile X mutation. And as I've already said, even before this was discovered, people got the hint that the amount of methyl donors you had affected the expression of this Fragile X chromosome. Meaning that this is clearly something that has to do with DNA methylation as well as this particular repeat length mutation. So we already learned, thankfully we went to the trouble of learning about repeat expansion in the last lecture, so I don't need to go over that again, but suffice it to say that trinucleotide repeats are unstable during mitosis and meiosis. So whenever a cell is synthesizing DNA, there is the opportunity there for the repeats to expand. In fact, making the condition worse the longer it hangs around in the gene pool. This can explain also why some people are carriers. You may have a number of repeats that's below the threshold to cause disease in you yourself, but it may cause disease in your offspring if the repeat happens to expand beyond a certain point. The number of repeats a parent has, just like in Huntington's, correlates with the rate of expansion in their offspring. So here is an example from Fu et al. After this repeat had been discovered and it became possible to measure objectively, you could count the number of repeats in the parental allele and plot that on the x-axis, and then plot the change in the offspring's allele on the y-axis. So if, for example, the parent has 80 repeats, then on average, the child will have 80 plus, let's say 20. So the child will have like 100 repeats. The rate of expansion in the child is positively correlated with the starting point that their parent gives you. So if you have 100 repeats from your mom, you might add an extra 40 on top of that. So the repeat length can expand considerably depending on the parental allele. This is the very exact same as how it works in Huntington's disease. Because longer repeats just tend to be less stable and tend to expand more readily. So let's get into the epigenetics of this a little bit. And now I'm kind of getting out of the historical, storytelling mode and going into just here's the facts of the matter, because we're in the modern times now and we don't need it's not quite as, it's a little bit less fun to talk about this as a detective mission now that we're in the modern day. But here's how it works. There is a gene called FMR1. This gene was discovered after this repeat was discovered people identified where the repeat was and then noticed there happened to be a gene very nearby. And Fragile X syndrome is caused by an expansion of this CGG repeat upstream of the FMR1 gene. So normal, healthy individuals would have between 6 and 50 repeats, and people with the full mutation would have over 200. In between, you would have sort of a partial mutation or I guess a premutation, and you may or may not have symptoms, but the odds are good that you might eventually pass it on to somebody in your family because of the way that repeat expansion works during meiosis and mitosis. So the gene was discovered downstream of this Fragile X causing mutation, and so the gene received the rather unfortunate name of the Fragile X mental retardation 1, because that's the syndrome it causes, and that's what the syndrome used to be called back in those days. So it produces the Fragile X mental retardation protein, FMRP. The name of that still is what it is. Like I said, it's just, there's a lot of things that are named in genetics that really kind of have bad sounds in our current understanding or our current ears. But kind of the way data is like stored and recorded, it's very tough to kind of retroactively change the name of things if you don't like the way they sound, because decades of scientific literature have been built up with this particular name. So we can just call it FMRP. There's kind of another funny story. Maybe I'll tell you about this in the obesity lecture. But one of the main genes that was linked to obesity was called FTO. It was discovered pretty much out of the blue by a GWAS survey. And FTO, purely coincidentally, based on research in mice, I think, was called FATSO. That was the name of the gene. And it was only because the size of the gene was really, really big. But people realized that would be a horrible name for a gene that was associated with obesity. So it was called fat mass and obesity associated. So it was given a better name at that point. So it's not impossible to rename genes, but it's challenging, and it requires a consortium of international people to get together and make all these decisions. So, it's one of those things, I guess. But we can just call it FMR1 and not think too much about it. The gene itself actually is kind of ironic, because what you actually... Well, anyways, I guess I'll get to it. So this is what causes it, essentially, is expansion of this CGG repeat. And the real secret to what actually causes the syndrome is not so much the expansion, although the expansion is relevant, but it's the methylation state of the expansion. Because, of course, CGs can be methylated, or CBGs can be methylated. We can represent that here with these lollipop-like things. A methylated CBG is a filled circle, and an unmethylated CBG is an empty circle. And you can see how the methylation state upstream here is, maybe methylated, maybe not. But in the full mutation, you tend to see very high levels of methylation here. All of the cytosines are fully methylated. And or what you might say hypermethylated, meaning more methylation than normal. And so you've got a ton of extra CPGs that aren't really supposed to be there with very high methylation levels. What this ultimately results in is the epigenetic silencing of the FMR1 gene. Normally the FMR1 gene is expressed, it's an important part of brain development, but in the case of Fragile X syndrome, its expression is silenced epigenetically by all of this extra methylation. This methylation causes the gene to not be expressed, and therefore whatever it normally does during brain development is not going to happen the regular way. So this really kind of answers the question that Howard Peebles raised in '81, because they noticed that the level of methionine in the medium seemed to correlate with the level of expression of this X chromosome deformity, and lo and behold, the reason for this is because the actual physical manifestation of Fragile X has to do with methylation. DNA methylation was not a topic that they thought very much about back in the 80s. It's very popular now, but it wasn't a huge thing back in those days. So we kind of answered that question for her, and I think it's pretty cool. So how does this methylation cause the fragility of X? We can see how it impacts the expression of this FMR1 gene. FMR1 gene, of course, being essential for brain development. You need it to be expressed. If it's not expressed, then you're not going to have normal brain development. That's simple enough, I suppose. The fragility itself is caused by the DNA around the FMR1 gene being highly constricted. So the chromatin, in other words, is in a very compact state. And as I've mentioned, epigenetic mechanisms like DNA methylation and like histone modification can regulate chromatin conformation. So heterochromatin, for example, heterochromatin is where gene expression, or genes in heterochromatin are not expressed very much, if at all. And these are areas with high levels of DNA methylation and low levels of histone acetylation. So there's kind of an epigenetic profile associated with these silent areas of the genome characterized by very high levels of methylation across the board. So of course, if you take an area of the genome that's not supposed to be heterochromatin, you add a mutation that fills it up with cytosines or CpGs, and then you add a whole bunch of methylation to those CpGs. In a sense, you're taking a part of the genome and converting it to something like heterochromatin, even when it's not supposed to be. This will change the three-dimensional structure of the chromatin and cause all kinds of physical problems with the DNA, as well as problems expressing the genes in that locus. And Coffey et al. In 1999 found that histone acetylation is depleted near the FMR1 promoter as well. So this is kind of showing this interaction between different epigenetic signals. DNA methylation also regulates histone acetylation because the enzymes that modify histones, in a sense, read out the methylation state of that DNA and then change the histone modifications to be consistent with it. So if you have a lot of methylation, that's an area of the gene or of the genome that doesn't want to be expressed. You can come in and remove a lot of the acetylation marks, or decrease the level of histone acetylation. That will further suppress gene expression. That's histone acetylation is one mark that can do that. It'll further suppress gene expression and cause the physical shape of the DNA to actually change. So according to the authors then, high levels of DNA methylation are actually causal. Histone deacetylation is a secondary effect, but the two of them work together to silence gene expression and condense the chromatin in that area. So many studies, so these studies, studies that try to look at this more closely, as a purely technical note, oftentimes take advantage of the fact that you can treat cultured cells with drugs like decitabine that inhibit de novo DNA methylation. And this actually might have made you think, like, well, if restricting methionine in cell cultures could fix the X chromosome or make it so it wasn't fragile anymore, couldn't we take carriers, or couldn't we take fetuses or embryos or whatever who we think might have fragile X and restrict their methyl donors so that they wouldn't develop it? And in a sense, that maybe could work. If you could somehow target your inhibition of DNA methylation to that exact fragile region, you actually might save quite a lot of damage. However, just putting a drug in or taking away all the methyl donors is not going to do the trick. Decitabine is a chemotherapeutic agent. You would use it if you had a very advanced type of cancer and there was nothing else that really could be done for you. It would not be at all useful as a prophylactic. It would cause severe birth defects and many other problems, so you can't use it in that context. But the dream, I guess, is there of somehow being able to target the de novo methylation happening at this CGG repeat and block it somehow. And if you could figure out how to do that, you likely would be able to prevent some of the worst features of Fragile X from developing. So it's always, there is a pathway here I think we could kind of work with, but it's a long way off from being able to work, to really put into practice, I guess. So what is FMR1? This is this Fragile X mental retardation protein gene. This is something that you actually want. So it's despite its name, you actually, if you want to have a healthy and normal brain, you actually want more of it, not less. Silencing it causes Fragile X. What does it do? Well, FMR1 is widely expressed in the brain. It's seen in both neurons and glia, so its expression is not limited to one cell type. In neurons, the protein is found in the dendrites and the synapses and the axons, so sort of the processes of the cell, but it's also found in the cell body. It's found kind of basically everywhere in the neurons. It's also found throughout the body, particularly in the testes. We don't really know why it's there. I guess a testicle scientist might know, but I don't particularly know that kind of thing. But it's found elsewhere in the body, as many genes are. Sometimes genes are expressed in the brain, but also some other part of the body. But in the 90s, it was found that FMRP is a RNA binding protein. So the suggestion then by Kandijian et al. Is that the FMR1 protein controls protein translation, because of course ribosomes are where mRNAs are converted into proteins. So if you have something that binds to RNA, what you might have on your hands is a protein that kind of fine-tunes protein synthesis and regulates the rate at which it happens. Maybe if you don't have it, your cells produce the wrong amount of protein, and that could be kind of problematic. And in fact, this is what we see. So in 2013, Chin et al. Found that if you knock out FMR1 in mice or in cell cultures, the rate of global protein synthesis increases in the hippocampus by between 15 and 20%. So that's a considerable amount of extra protein being made in the brain that's not normally ordered. Typically, if your cells are to be in good homeostatic condition, the rate of protein synthesis should be proportional to what's needed by the cell. You know, it's a bit like if you're ordering pizza or something, you're ordering an amount of pizza appropriate to your needs. If you're all by yourself, you might order one pizza. If you open the door and a buddy shows up and he's got a truckload of pizzas for you, that's too many pizzas. You can't use that many pizzas. You know, you can't even fit it in your freezer. Cells are kind of the same way. They order a certain amount of proteins. They transcribe mRNA at levels proportional to the amount of the protein that they need. So proteins they need a lot of, they'll make a lot of mRNAs for that protein. For proteins they don't need very many, they'll make fewer mRNAs, and the translation rate is kind of proportional to that. So there's an appropriate amount for all these things, there's an appropriate level for all of these things, and it's fine-tuned by a whole range of mechanisms. And it turns out that this Fragile X mental retardation protein is probably doing that. It's probably controlling the rate of protein synthesis. It also seems to control stimulus-induced protein synthesis. So there's a level of baseline synthesis in the brain. You need proteins to live and to build up your, to run your metabolism and to build up your cells. But in response to a stimulus, for example, if you need to learn something, you need to have protein synthesis to build new molecules on your synapses and to build out new structures and to grow out your dendrites and all the other kinds of things that neurons do when they're learning or when they're exposed to stimulus. You need protein synthesis. You might need new NMDA receptors or AMPA receptors or whatever it is. So the FMRP also seems to be involved in that. So what essentially is happening, or the conclusion then, is that FMR1 is a gene that codes for a translational suppressor. It's a protein, the FMR1 codes for a protein, FMRP, that slows down the rate or controls the rate of protein translation, and thus the kind of overall function of the cell. And if you don't have it, that's not good. So here's the diagram made by Richter et al. That kind of shows this. Normally, so here's an example of what protein synthesis actually looks like. So you've got your RNA here with the poly A tail. This line here is your RNA, or your mRNA to be precise. The ribosomes are built up of multiple subunits, but they're these kind of snowman-looking things. They'll clamp onto the RNA at the translation start site. The AUG is where the TSS is. That codon specifies the transcription start site. It's ATG in the genes, but it's AUG in RNA. They'll clamp on and they'll travel down the line, spitting out the peptide as it goes. And so the peptide is longer and longer as you add more and more amino acids to it. And FMRP, this Fragile X mental retardation protein, binds to the ribosome and somehow regulates the rate at which this happens. How it does it, we don't exactly know yet. And in Fragile X, it's really just the fact that the protein is not there, and the rate at which this happens is now under less control. The FMRP, I guess they're arguing, acts kind of like a roadblock, slowing down ribosomes as they travel down the length of the mRNA, and thus slowing the rate of polypeptide elongation. So if it's missing, the rate of protein translation is increased beyond the normal rate. You end up with more copies of that protein floating around, and that can be bad in all kinds of different ways. So if you knock out the FMR1 gene in mice, you'll see a phenotype that is fairly reminiscent of Fragile X syndrome in humans. The Fragile X, or the FMR1 knockout mice have enhanced long-term depression induced by the metabotropic glutamate receptors. I'm assuming that you must have heard about metabotropic glutamate receptors in another class. I'm not really qualified to teach you the neurophysiology of it, but This is one type of synaptic plasticity enabled by a type of glutamate a glutamate receptor that's not the AMPA or the NMDA types or the kainate. It's a different kind of a G protein coupled glutamate receptor. Anyway, it's a type of synaptic plasticity that is actually enhanced in these knockouts. This is a type of synaptic plasticity that depends on protein synthesis, and so the normal mechanisms of synaptic plasticity, to the extent that they depend on protein synthesis, are disturbed in people who have Fragile X syndrome. More proteins is not always better, even though the protein is what enables this kind of long-term depression. Your brain is expecting to work a certain way, and having one aspect of its synaptic plasticity working at a different rate than normal is going to produce pretty bad results altogether. Oh, I forgot to mention, if you don't know what macroorchidism is, it means large testicles. Orchid means testicle. So if you ever the surgery to remove to castrate animals is sometimes called an orchidectomy, and that's because of the word orchid means testicles. And the flower, if you ever pull an orchid flower up by the roots, you'll see root structures that look very testicular in nature. So that's the root of that word. Biologists are such classy people. Anyway, the FMR1 gene also have decreased, or the FMR1 knockout mice rather, have decreased long-term potentiation as well as decreased GABA signaling. So a whole host of problems with the way their brain works caused by this mutation. I think we can't read into it too much in the context of mice, but suffice it to say, if you take away this gene, you end up with a lot of neurological problems that could explain many of the traits and phenotypes we see in the condition itself. So we got a question here from Nathalie. So FMRP could be considered a speed bump in the road to slow down protein synthesis. Yes, I think that's a great way of putting it, actually. So the thinking is that it slows down the rate of protein synthesis by physically interacting with the ribosomes as they travel down the mRNA. So it is physically blocking them somehow or slowing them down. Why that would need to happen, I'm not entirely sure, but that is physically what's going on. So it is sort of a speed bump in that process. But anyway, you can take all of these findings from mice studies, and believe me, you could go on and on about this. I'm only giving you the slightest taste of the literature, but you can kind of go on and on. And what seems to be the case is that the deficits seen in Fragile X syndrome result from changes at the level of the synapse. The wrong amount of protein synthesis is happening, and as a consequence, the normal mechanisms of synaptic plasticity as well as neural development do not work as intended. And this leads you to have a whole range of cognitive impairments as well as problems with the neurogenesis and other kinds of things. So I think that's actually my last slide, folks. So once again, we're done a little bit earlier. That's good for everybody. Well, I guess I'll just summarize verbally a little bit because I guess I never put a summary slide in here. But what I hope I've shown you, or at least my goal in this class, was to show you how you can move from a pedigree and an observation that a certain type of cognitive impairment runs in families along the female line, and you can bring it all the way down to the level of synaptic function in mouse knockout models. And as you go, step by step, you can uncover different hints as to how the disease is caused. This is the beauty, by the way, of these kind of monogenetic trinucleotide repeat conditions, is that there's just one thing that causes it. And so it's very easy to figure out what that one thing was. I shouldn't say it's very easy. It was actually very challenging for the people doing the work. But in retrospect, you can look at it, and it's very simple to think, okay, there's one place in the genome where if there's a mutation there, it causes this hypermethylation. The hypermethylation affects the structure of the chromatin, and it affects the expression of the gene. This causes all these problems that we can model in mice. Bob's your uncle. So that's the beauty of these simpler disorders. We're not going to have, we're going to have to go back to the more complicated polygenic conditions starting on Wednesday, but you can see how it works anyway. And I hope I've given you a sense of the progression from observation all the way to the latest findings. So I'll leave it there, my friends. Hopefully you learned something today and found it interesting. As always, be sure to finish your assignment. I guess that's due today. study for your exam, and please let us know if you have any questions about things or if you have any issues that you need help with. That's what we're here for. The exam is not going to be any easier than the last one, although I think it might feel as though it's easier because I think you're a bit more prepared for it this time. So hopefully that will inspire you to study hard and work hard. And so enjoy, my friends. We'll see you on Wednesday, and if you feel like it, we'll also see you on office hours on Thursday. Have a good rest of your week.